The title today is The Latest Developments in Cancer Research, and we're extraordinarily fortunate to have two eminent researchers from the Centre for Cancer Biology. And the Centre for Cancer Biology is an internationally recognised medical research centre that investigates the biology underpinning cancer and translates this knowledge to provide better outcomes for cancer patients. The Centre for Cancer Biology, or CCB, is a strong alliance between SA Pathology and the University of South Australia, and it harnesses the strengths of both partners to chart the path from scientific discovery to clinical implementation. Our first speaker today, not only a member of the CCB, but also the director of the Centre for Cancer Biology, is Professor Natasha Harvey. It's wonderful to have her here, and she's also the head of the Lymphatic Development Laboratory within the Centre for Cancer Biology. Natasha's research is directed towards understanding how the lymphatic vasculature is constructed during development and how this process goes wrong in conditions such as cancer, vascular malformations, and lymphedema. In the lymphatic system, for example, groundbreaking work by Natasha has highlighted the importance of a nuclear protein which regulates the expression of genes, called GATA2, in lymphatic valve development and in lymphatic valve maintenance. It's called a transcription factor, and it transcribes the right message, to use my lay terms, and when the encoding goes wrong or the process goes awry, then conditions such as lymphedema result. Natasha is an ARC Future Fellow, an Australian Research Council Future Fellow. Her work has been published in a number of leading international journals, and journals like Nature Genetics and the Journal of Clinical Investigation and Science Translation Medicine epitomise to me the commitment both of the CCB and Natasha to chart the path from scientific discovery to clinical implementation. Please join me in welcoming Natasha Harvey to speak to us. Thank you.
Thank you, Ruth, for that very generous introduction. It's a pleasure to be with you here today. We're going to talk about some of the research that's very much in progress at the Centre for Cancer Biology, which is located right across North Terrace from this building, in the Bradley building. It's an amazing place to work, and there's some amazing research going on there. So, as Ruth mentioned to you, my research program is focused on lymphatic vessels. So I'm going to tell you about why lymphatic vessels are important and how they go wrong in human disease and what happens to them as we age. And then my colleague Yisim Goodall is going to talk to you after the break about one of the very exciting projects in her laboratory that is focused on developing new cancer therapeutics for breast cancer. So this is a really exciting opportunity to talk to you about what we're doing, but please don't hesitate to ask us any questions you might have about our research programs. So here on the screen, you can see what lymphatics look like in the skin of a mouse. So this is an image we've taken in a high-resolution microscope, and you can see here the lymphatic vessels are made up of these cells that have red nuclei, and that's one of their distinguishing features. That's how we can tell that they are lymphatics in identity. But before I talk to you about my own research program, I wanted to introduce to you the Centre for Cancer Biology and tell you a bit broadly about what our research teams are doing. So, as Ruth mentioned, CCB is an alliance between SA Pathology and the University of South Australia, and this enables us to take advantage of the very strong clinical links of SA Pathology and the very strong research capacity of the university to really chart new ground in cancer research. So we are made up of 19 research laboratories, and they're all listed here, and you can see that the gene regulation laboratory, for example, is a very specific name, but broadly our research programs cover many different aspects of cancer, and they range from understanding the very fundamental genetics and cell biology that go wrong to cause cancer, right through to very translational applications. And this is right through to the development of new therapeutics that are undergoing clinical trials. For example, in the research program led by Professor Michael Brown, who's an oncologist at the Royal Adelaide Hospital. So we span all aspects from discovery to translation, which is a really exciting place to be. And our links with clinicians enable us to understand what the clinical problems are and then to imply or put into practice any therapeutics that are developed. So it's a really fantastic and very stimulating research centre to work in. So the names of the laboratories, as I mentioned, are very specific, but broadly we cover many different types of cancer and many different aspects of cancer. So today you'll hear from Yisim about her breast cancer research program. We have many labs that focus on breast cancer research. We also have research teams focused on leukemia and many different types of leukemia, on brain cancer, on pancreatic cancer, and on melanoma. So there's a lot of exciting work going on at the centre, and we'll be happy to answer any questions you might have about particular research programs later on as well. So lymphatic vessels, what are they and why are they important to our bodies and our well-being? What astonishes me is that lymphatic vessels were first described by the ancient Greeks. There are records of this around 300 BC, and I consider this particularly astonishing because lymphatic vessels are not as easy to recognise as blood vessels are. Blood vessels carry our red blood cells around our bodies together with oxygen and nutrients under high pressure, and that's why they're very easy to see. But lymphatic vessels carry a colourless liquid around our body and carry white blood cells, which are really important for our immune responses. So they've been much harder to spot than blood vessels, and as a result, until about the last 20 years, have received much less attention from the research perspective than their blood vessel counterparts have. But I think some of these drawings here on this slide are quite astonishing because they depict what anatomists hundreds of years ago now could recognise. So these are lymphatic vessels that drain the human intestine of fats and lipids. These are the large lymphatic vessels in the centre of our bodies that carry the highest lymph load in the body. And these are the lymphatic vessels, the very superficial lymphatic vessels in the skin as drawn by Sappi more than 200 years ago now. But what are the functions of lymphatic vessels? They're very important for fluid homeostasis. So, as I mentioned, our blood vessels carry blood under high pressure, and that means that the liquid part of the blood comes out of blood vessels every day. So one of the most important jobs of lymphatic vessels is to pick up that fluid and take it back to the bloodstream. Along the way, that lymphatic fluid is drained through lymph nodes, and you're probably familiar with lymph nodes. You'll often feel that they might be swollen when you have an infection, and that's because there's a lot of immune cell activity going on to respond and fight that infection. So the lymphatics drain their contents through lymph nodes, and that means that all of the fluid that goes through that system is presented to our immune system for surveillance and for the ability to mount an effective immune response. So lymphatics are really important for our immunity. And as I mentioned, they're also very important in lipid metabolism, in absorbing the lipids from our diet and taking them forward for further processing. So my lab is really interested in understanding how lymphatic vessels are built during development and how this process goes wrong in human vascular diseases like vascular malformations and lymphedema. And we're particularly interested in understanding how the cells within lymphatic vessels are different from those that make up the blood vessels and how they are transcriptionally programmed. That is, how do we make that cell the identity that it is to build a lymphatic vessel? And I'll talk to you more about what we study with regard to transcription factors that really control the identity of the cells as they develop. I'll also talk to you a lot more about valves. Valves within lymphatic vessels are really similar to valves within our veins and valves within our hearts. And they're really important to making sure that the fluid that's picked up by the lymphatic vessels is transported in one direction back to the bloodstream. So the lymph that's picked up in the periphery of our tissues is drained via larger and larger lymphatics, including the thoracic duct, back to the bloodstream at the junction of the large lymphatic vessels, the thoracic ducts, with the jugular veins. And this is the point at which lymph fluid goes back into the bloodstream. So you can imagine if this process of fluid return doesn't happen, that fluid accumulation is the end result. And that is in fact what goes wrong in diseases like lymphedema. So this is cartoon format, looking at the cells that make up lymphatic vessels. And this is what those vessels actually look like in the skin of an adult mouse. So here we've labelled these initial collecting vessels into their initial counterparts, which pick up the fluid and drain it into the collecting vessels. And the collecting vessels are where the valves are located. And these collecting vessels are really specialised to pump and to make sure that lymph gets returned to the bloodstream. So what happens if this process goes wrong? If lymphatics aren't built properly during development, and this is fetal development in utero, a number of conditions can result. One of those is vascular malformations or lymphatic vessel anomalies, and this is an example of one of those conditions here. So this is a tracer, if you like, where we've injected a labelled product and looked at how effectively that's returned to the bloodstream via the lymphatics. So what you might appreciate here is this labelled white tracer is coming via these large lymphatics back up to the thoracic duct to the point where it meets the bloodstream. But in this patient who has a central conducting lymphatic anomaly, this tracer is not effectively transported. And what you can see, in fact, is the lymphatics haven't formed properly. They're very large and distended. And the result of that is that lymph can't be returned properly to the bloodstream. So this causes a major problem because that fluid accumulates in tissues. And it's particularly damaging when it accumulates in utero, in the developing fetus, in tissues including the heart and lungs, because it disrupts cardiovascular function. These are two other examples of what happens when the lymphatics don't work properly because there are problems with their structure. This is the scenario called lymphedema, and in this case this is primary or inherited lymphedema, and in this case this is secondary lymphedema. So this child, you might notice this limb is dramatically swollen. This is not simply just fluid, but it's the duration of this event has led to the formation or accumulation of adipose tissue, as well as fibrosis and chronic inflammation. We don't have any good treatments for lymphedema besides bandaging or compression and massage. So one of the goals of our research program is to identify ways that we can promote the growth of new lymphatic vessels or repair them and promote their function to get better treatments for lymphedema patients. While this form of primary lymphedema is relatively rare, secondary lymphedema is much more common, and this can be a major problem for cancer patients who undergo surgery to to remove their lymph nodes when cancer has spread. And by taking away that lymph node, that interrupts the lymphatic transport function. So secondary lymphedema can affect up to 20% of patients after surgery for cancer. So again, we'd really like to be able to provide effective therapeutics for this kind of scenario. But lymphatic vessels are implicated in a wide array of additional pathologies in humans. We've known for a long time that cancer cells can spread to other parts of the body by entering the lymphatics. And in fact, more recent work showed that some types of cancer actually produce factors that promote the growth of new lymphatics towards the tumour, and this provides a ready access point for the cancer cells to enter lymphatics and spread to the draining or local lymph nodes in close resonance to that tumour. From there, cancer cells can actually access the blood vasculature and thereby spread to more distant parts of the body. So this is a case where for a long time we had associated the growth of new lymphatics around cancer cells as a potentially damaging impact. But a large body now of more recent work in the field is showing that actually the growth of lymphatics around a tumour can have really positive outcomes as well. And that's because lymphatic vessels carry immune cells, and these immune cells can have a really beneficial effect in fighting that cancer, particularly with the advent of new immunotherapies. So I think what you might recognise here is that we have to understand the scenario of each type of cancer and the way that lymphatic vessels grow around that cancer environment to understand whether promoting the growth of cancer of lymphatic vessels would be beneficial or whether we need to identify ways to stop cancer cells from getting into lymphatics and spreading to other parts of the body. Because of their role in immunity, the growth of lymphatic vessels is promoted in a wide range of settings of inflammation. And I show here two examples of that today. In red, you might appreciate the growth of new lymphatic vessels that has occurred here in a mouse model of infection. And again, this is advantageous to being able to mount an effective immune response to fight that infection. While in the setting of organ transplant, the growth of new lymphatics enables an immune response to be mounted that's not effective and that can be associated with transplant rejection. So again, we really need to understand the context that we're looking at in any particular disease to understand how modulating lymphatic vessel growth could be beneficial. Because of their role in lipid absorption, the lymphatics have been associated with obesity in mouse models, and numerous research teams are now looking at whether lymphatic vessel dysfunction might be associated with obesity in the human population, and if so, what are the contents within lymph that are able to drive obesity. Lymphatic vessels in our lungs are really important for first inflation of the lungs at birth, and this is a mouse model where the lymphatics don't grow properly and it results in the death of these mutant mice. A really exciting area of research in our field is focused on the lymphatic vessels that grow around our hearts. So this is a heart, an embryonic heart of a mouse, in fact, and these yellow vessels are the lymphatic vessels that grow to cover the heart. And again, exciting research by a number of teams internationally is now looking at the fact that promoting lymphatic vessel growth and function can lead to promotion of cardiac repair after heart attack. And another exciting recent emergence in our field has been the recognition of the importance of the lymphatics that drain our cerebrospinal fluid. So these lymphatics don't actually enter the brain itself, but they cover the surfaces of the brain. And they're very important for monitoring the contents of CSF and for removing the waste products. So the degeneration of lymphatic vessels has been associated with neuroinflammatory diseases like Parkinson's disease and Alzheimer's disease. And recent work by a number of teams has shown that if we promote the growth of new lymphatics in and around the brain, that we can stop or help to prevent that neurodegeneration. The good news is there's a lot of really exciting work going on in the field of lymphatic vessel biology research, which will, I'm sure, lead to new treatments for a really wide array of diseases. The more disappointing news is, like many aspects of our physiology, lymphatic vessels do degenerate with ageing. And so by identifying ways that we can really promote and boost their maintenance, their function, and even potentially their repair, we hope to have a wide impact on health generally. So I'm going to tell you about 3 short vignettes, 3 projects currently ongoing in my research team, and they all focus on identifying what the genetic events are that go wrong in human lymphatic disease, and then understanding at the very molecular level how those genetic events cause problems with lymphatic vessel development. And of course, this is fascinating. It's a fascinating question, but it's also a question that we need to answer if we want to be able to develop effective and targeted new therapeutics for these diseases. So the first disease I'm going to talk to you about is a syndrome called Emberger syndrome. It results in primary lymphedema, so the swelling of the limbs of these patients, as well as a predisposition to myelodysplasia syndrome and acute myeloid leukemia. And this is because GATA2 plays important roles in more than one tissue. So we have known for a long time that GATA2 plays important roles in the production of blood cells during development, but the finding that GATA2 mutations cause this syndrome that's characterised by lymphedema actually told us that GATA2 must also be doing something important in lymphatic vessels. And our job as developmental biologists and geneticists was to understand what that was. So the first thing that we did was to map where GATA2 is in lymphatic vessels. And this is a lymphatic vessel labelled here by the red cell nuclei. This is the area in the cell where our DNA resides. And what we could find was that we saw 2 places where we saw really high levels of GATA2. One is in these scattered cells in the skin, which are cells of the immune system, so that fits with GATA2's role in blood cells. And the other place we saw lots of GATA2 was this really restricted population of cells, which corresponds to this, the cells here at this vessel branch point, which happen to be the cells that build the first valves in lymphatic vessels. So here we come back to valves. The other place in lymphatic vessels where we saw high levels of GATA2 was another specialised valve population. And these are the valves called lymphovenous valves, which are located at the point at which lymph is returned to the bloodstream. So this valve lets lymph come back into the veins, but it stops blood from entering the lymphatics. So if we asked the question, if GATA2 is really present at high levels in valves, might it be important for building valves or maintaining valve function? And what do we know about that process? So as I mentioned, just like valves in veins, hearts and pipes, the valves in lymphatics are really important for making sure that fluid is returned effectively to the bloodstream. It can only go in one way, essentially. So during development, the first sign we have that valves are starting to form are these cells at vessel branch points, which is where we saw GATA2, which dial up their levels of a number of different markers. These are all transcription factors like GATA2 is. These cells then go on to remodel their shape loosen their junctions to the cells they're next to and invade into the lumen of the vessel. And as they do that, they build leaflets, which are endothelial cells, that is the cells that make up vessels, but also they are provided with some structural support, some mechanical support along the way that's really important for valve function. So we asked, is GARDA2 important for building valves? And to cut many years of work very short, the answer is yes, they are. And GARDA2 is important for building the valves in the lymphovenous valve, and it's also important for building the valves in the collecting lymphatic vessels. So this is what valves during development normally look like. We can see them because they have high levels of these transcriptional regulators, but in the absence of GARDA2, these valves never form. And in fact, if we allow development to progress and then interfere with GARDA2 function, we've done this genetically, those valves start to disintegrate. So GARDA2 is really important in building valves. It's a transcription factor, that's a molecular switch that binds to DNA to turn genes on or off. So our next question has been, how does GARDA2 control the process of valve development by regulating the turning on or off of a whole suite of genes? And again, I'll cut to the story short to tell you that one of the genes we found that's regulated by GARDA2 that really jumped out to us as being important was a gene called FAT4. And it jumped out as important because FAT4 mutations, that is mistakes in the gene that encodes for FAT4, had just been identified as causative of another primary lymphedema syndrome called Hennekam syndrome. And this is an example of the kind of tissue swelling that can occur in these patients. What is FAT4? It's interestingly not a transcription factor. It's a really huge molecule that resides on the cell surface, and we think it's really important for organising cells within tissues, for binding cells to one another. And that had been shown by a number of research teams that FAT4 has important roles in organising how cells are put together in tissues to form organised patterns. And it had also been implicated in a really important signalling pathway, a growth pathway that goes wrong in cancer, called the Hippo pathway. So our work, again, set out to look at where FAT4 was in lymphatic vessels and to understand what the impact of interrupting FAT4 function is on lymphatic development. And again, we've used a genetic model and shown that when we remove the activity of FAT4, the lymphatics in the skin of these mice look really misshapen. So they're very large and unlikely to be effective or functional. Looking very closely at valves, because we knew GARDA2 had an important role in valves, what we saw was these normal valves in a wild-type mouse, but in a FAT4 mutant mouse, we saw far fewer valves, and the valves were less well formed. So we think that patients with FAT4 mutations, again, develop lymphedema because their valves aren't built properly and their lymphatics can't return fluid effectively to the bloodstream. In fact, further work in the lab has shown that FAT4 is really important for organising the shape of cells within lymphatic vessels. So what we could see was that in normal lymphatic vessels, the cells are really beautifully elongated into tubes as they're built, but when we remove FAT4 function, the cells remained really round and enlarged. So the end outcome of that is that instead of building nice, tightly, well-plumbed vessels, we're actually generating big bulbous vessels that really don't work very well. So the third example of recent projects that are still very much in progress in our lab is a really exciting international collaboration, again working with geneticists both here in Adelaide but also in Europe and the US, that identified in another lymphatic disease called central conducting lymphatic anomaly mutations in a gene called MDFIC. And to be honest, when we first found mutations in this gene, I knew nothing about MDFIC. So this is how serendipity takes us down different research pathways. So in this case, this gene was discovered because our colleague Hamish Scott wanted to understand the genetic basis of stillbirth. So for many couples who lose children to miscarriage or stillbirth, there's no definitive answer as to why that occurred. And Hamish really wanted to find the genetic answer underlying this. In this particular family, first identified in Adelaide, they had lost 2 pregnancies where the fetus had developed the condition called hydrops. That is the accumulation of fluid in the fetus, which is particularly damaging to heart and lung function. And Hamish's team identified that the gene event that had gone wrong in these patients was MDFIC. So this was the Adelaide family. There were 2 affected pregnancies where the fetus had 2 copies of MDFIC, so both copies of the MDFIC gene contained mutations that resulted in disease. And by reaching out to colleagues around the world, we in fact found another 6 families who had children or pregnancies affected by mutations in this same gene. And this really gives us the confidence to say, well, it's definitely the MDFIC gene that's the cause of this disease. So while some families actually, unfortunately, lost their children through stillbirth, the children who survived had the constant accumulation of fluid in their lungs and hearts, interrupting cardiovascular function, as well as lymphedema. So a very severe disease, and a disease that we really hope we can develop therapeutics for by understanding the biology that really underlies this disease. So what does MDFIC do? It's really important, again, for building lymphatic vessels during development. And these are some images that were taken by our clinical colleagues in the US, and they show the kind of misrouting or reflux of lymph in this affected child and the lymphedema that is a result of that. So this time, instead of disrupting the function of the gene by removing it entirely, we generated a mouse model that carries the same genetic event that is found in patients. And by doing this, we were able to see that the mice that carried 2 mutated copies of this gene, that is the same scenario that we found in patients, they actually die within the first couple of weeks after birth, and they die with this accumulation of lymph. It looks milky here because the pups are still suckling milk from their mothers, and that's absorbed via the lymphatics. That's the lipid and fat connection. The entire thoracic cage gets filled with lymph, and this stops the heart and lungs from working properly. So that's why these mice die. So how does MDFIC control the process of lymphatic vessel development or function? What we found again was that the lymphatic vessels in these mutant mice are really big compared to the well-formed lymphatics of their wild-type counterparts, and these images are taken in the thoracic cavity, but in fact the lymphatics were malformed in every part of the body that we've looked at. And this is an example of the collecting lymphatics in the digestive tract. These drain the lipids from the gut to the mesenteric lymph nodes. And again here you see the formation of normal valves in a wild-type mouse, but in a mutant mouse these valves don't form at all. So MDFIC just like GATA2, just like FAT4, is really important for building valves. So I think that gives you an insight into how important the process of building valves properly is and maintaining their function for effective lymphatic function. So I'll just summarise there and tell you what we're currently working on. Lymphatic vessels play crucial roles in health and disease, really in a widespread manner, by virtue of the fact that they're important for fluid homeostasis, for our immunity, for being able to mount good immune responses, and for lipid absorption. And I hope what I've illustrated is that understanding the genetics of human disease provides really deep insight into the cause of disease and the cellular and molecular events that go wrong, and enables the development of targeted therapeutics to be progressed. So by understanding really the nuts and bolts of what goes wrong in disease, we can work out ways to overcome how those events have gone wrong. Lymphatic vessel integrity and function unfortunately decline with ageing, but we hope that our research will identify ways in which we can optimise lymphatic vessel health and function and therefore have widespread health benefits. So if anybody has questions that you don't have a chance to talk to me about today, you're welcome to email me anytime on this email address, and in the meantime I'll be happy to answer questions right now. Thank you.
Thank you very much, Natasha. We have plenty of time for questions before we break for afternoon tea, so please. One minute, we'll just bring the mic so we can.
Thank you. In one of the FAT4 overheads, it said something can be allelic to. What does allelic mean?
Yes, so in fact, in the case of Hennecomb syndrome, mutations in FAT4 have been shown to cause Hennecomb syndrome, but mutations in another 2 genes have also been shown to cause the same disease. So that allelic means the allele of the DNA that they're on, and in this case there can be multiple genetic causes for the same disease. And that indicates to us that while those genes are distinct, they are likely part of the same pathway, so that no matter what part of the pathway we hit, we get the same end result.
As an ageing person, I'm fascinated by the whole thing, thank you. And lymphatic vessel integrity and function decline with ageing. Are there any indications about what an ageing person can do to maximise the wellness of your lymphatics?
Yeah, I think exercise is really important because the muscles around our lymphatics are not as strong as the muscles around our blood vessels, and they are actually assisted a lot by the movement of our muscles, the muscles in our legs, also by our breathing and respiration, and by the pulsation of arteries. So exercising and getting muscles working is really important. And in the case where that can't happen, for example, you might notice your legs swelling on a plane or when you're sitting down for a long time, then massage can be really impactful in helping lymphatic function.
What actively drives the lymphatic fluid around the body? Like clearly with a heart, it's one central arrangement.
Yeah.
Is it a kind of a distributed heart, or there just relies on muscular and arterial?
So
pulsations?
the muscles around collecting lymphatics are important for contracting to help the lymph pump, and then that's the muscle cells, but then our larger skeletal muscles around those are also important in doing that. The lymphatics that actually pick up the fluid are really specialised, and instead of being the cells within those vessels being really tightly zipped together, they're buttoned together and they're anchored into the tissue by very elastic, rich filaments. And that enables that part of the lymphatic vasculature to sense changes in fluid accumulation, and they stretch open to allow gaps between the cells to open, and that enables lymph to go in. So that's almost like negative pressure to go in, and then the collecting lymphatics contract to pump the lymph back.
Yeah, thank you. The surgical response to cancer is usually, if in doubt, cut it out, which has resulted in a lot of lymph nodes being removed. In what way has your research informed any modification to that approach?
So not my research personally, but research in the field has really led to the development of mapping approaches. So instead of removing all lymph nodes, usually via dye injection techniques, the routes that are drained from particular areas are mapped, and so that only those lymph nodes are removed, leaving as many healthy lymph nodes intact as possible. So that's certainly an approach taken by surgeons to try and minimise the damage to lymphatics.
Congratulations. These genes are obviously involved in developmental biology. Where are FAT4 and MYFIC expressed outside lymphatic system?
Yeah, that's a very interesting question, and you've hit the nail on the head. All of these genes are present in other tissues in addition to the lymphatics. For example, in the case of FAT4, it's also present in the brain and the kidneys and the heart, where it plays very important roles, and that fits with some of the other characteristics exhibited by these patients. So there are problems with neural development, some problems with heart function, and problems with kidney function, in addition to the lymphatic problem. And MDFIC, similarly, we see really high levels of MDFIC. We haven't been able to understand what it does in a tissue selective way yet. We can do that genetically, and it's on our list to do, but it's also present in the lungs and the kidneys. So we're interested to see what its roles there are.
Thank you. Thank you for your talk. There has been some form of treatment for lymphedema to do with ultrasound. You didn't actually mention that, so possibly that's not effective. But if it is, what would be the mode of action?
I do understand that there's actually laser treatment available as well, cold lasers, and I think this can help to break down the tissue around lymphatic vessels to enable them to work better. So one of the other aspects of both ageing and a number of different pathologies is that the tissue around our vessels can become quite stiff and fibrotic, and that can impact how well the vessels are able to work. So I believe laser therapy works via that mechanism.
Yes, I am familiar, and I think the background to that question, laser treatment in lymphedema after a resection of the axillary nodes in breast cancer. But I wondered if that might be something you try on the mice on occasion.
We haven't yet, Ruth, we haven't. Yeah.
It's fascinating.
Yeah.
Any other questions?
There's one up the back there.
And right up the back.
Thank you. Just a question. A large number of women suffer edema and a breakdown of the lymphatic system in the last trimester of their pregnancy. Is there some research and understanding about that, or is that a question for the future?
It's a very interesting question. I can say that I personally suffered from a lot of fluid accumulation in my ankles when I was pregnant with twins. I think there are likely complicating factors. The simplest explanation is that the lymphatics, and particularly these large central lymphatics in our bodies, the ducts, are compressed simply because of a space issue, which makes lymph return to the bloodstream less effective. However, we do understand that there are hormonal influences on the lymphatic vessel function. We don't understand exactly what they are yet, but we know that some forms of lymphedema are more prominent in women or onset coincident with major changes in hormone levels. So there's a lot to be done, in fact, in that space, understanding how they regulate lymphatic function.
Thanks very much, Jay. Reflecting on what it's doing for your lymphatics.
Yeah.
Thank you very much for a very fascinating lecture. I have two brief questions. One is, what is the longevity of those unfortunate children with lymphedema? And second, would you regard gene editing as a likely answer to some of these problems?
So, to answer your second question first, gene editing is a very active area of research, not necessarily in our field, but for the treatment of a wide variety of genetic diseases. With regard to the longevity of children that have been part of our study, we have children that are very young, through to one child who's reached the age of 20. Unfortunately, some others have died before that time because the disease is very severe, and it can result in these children being highly susceptible to infection that they're not able to effectively mount a good immune response to. So unfortunately, this is a case of a very severe disease. What we really hope is by generating a mouse model that is a very good model of the human disease, we have identified some signalling pathways that are going wrong in those mice, and what we want to start now are some trials to look at whether by blocking those signalling pathways we might be able to treat the mice, and if that's the case, that's a good basis to work from to transition into humans. So we hope that we can develop some therapeutics that will help.
I've got non-Hodgkin's lymphoma, and I had chemotherapy which is put into the blood system. Does that make its way into the lymphatic vessels too, do you know? Or?
So it's the lymphatic vessels are like a highway that our immune cells constantly circulate through. So it's definitely possible that your lymphocytes have been moving through your lymphatics and that your lymph nodes might have been affected. So there's a very close relationship there.
Thank you very much for all those questions, but particularly thank you, Natasha. That's been quite a revelation, just how important lymphatic development, system development and function is to virtually every organ in the body. It's been terrific.
Great.
Thank you so much.
Thank you,
It's been
Ruth.
wonderful. We'll break for afternoon tea. It'll be served outside, and then we'll return here again in a quarter of an hour, and look forward to our next speaker. Thank you very much. Welcome back, everybody. Now we're going to hear from our second eminent researcher from the Centre for Cancer Biology, and that is Professor Yaseem Q. Goodall. Yaseem leads the cell signalling laboratory at the CCB, and to my mind, her research also epitomises so well the aim or focus of the Centre for Cancer Biology, namely to investigate the biology underpinning cancer and then translating this knowledge to provide better outcomes for cancer patients. Yaseem is committed to finding better treatments for the most aggressive types of cancer, like neuroblastoma in children and triple-negative breast cancer. As a consequence, her research focuses on the discovery of new biomarkers and therapeutic targets for these challenging cancers. And it seeks, too, to identify the mechanisms which underlie therapy resistance. Please join me in welcoming Professor Yaseem Q. Goodall to speak to us.
Good afternoon, everyone. So, firstly, thank you, Ruth, for the kind introduction. And secondly, thank you to all of you for coming down despite the inclement weather to listen to our talks. So, let me go to the first slide. All right. So my laboratory actually focuses on two cancers that are on two ends of the aging spectrum. So neuroblastoma is a childhood cancer, and the cancer actually develops when the baby is still inside the womb. And essentially, when they're born, they already have a cancer, even though it's not detectable usually at the time. However, and the other cancer that we study is, as mentioned by Ruth, triple-negative breast cancer, which is on the other side of the spectrum of middle-aged women. So since this is a successful aging forum, I've decided to focus on some of our work on triple-negative breast cancer and take you along the journey of discovery to trying to translate to finding better cures for this aggressive cancer. So firstly, breast cancer. Breast cancer is the second most highly diagnosed cancer in the world. And if you go to the Breast Cancer Network Australia website, you'll find something that looks like this with all the statistics. So in 2022 alone, over 20,000 women and a few men, so even men do get breast cancer, will be diagnosed with breast cancer. And of these, about over 3,000 of them will die just in the 1 year alone. And this represents about 15% of cancer-related deaths per year. Now, of the patients who get breast cancer, only 5% of these patients have a hereditary form of breast cancer with a known genetic mutation that they've inherited from their parents. So the remainder, so BRCA1 is a genetic mutation that a lot of people know well. BRCA1 mutations lead to aggressive ovarian cancers or aggressive breast cancers, and many of you would probably have heard the story of the actor Angelina Jolie, who inherited BRCA1 from her mother who had breast cancer, and she actually took the drastic step of having a double mastectomy so she wouldn't have the chance to get this aggressive breast cancer, which is difficult to treat. Now, for the 95% of patients who get breast cancer, the risks and causes are not really known. Some of it's lifestyle, some of it is environmental causes, but by and large, the biggest risk factor for breast cancer is age. So if you look at this graph here, which charts the age groups and the percent of new cases of breast cancer, you'll see that when you start hitting the age of 50, your risk of getting breast cancer increases. And this risk remains high until you hit your mid-80s. And then after that, as you get older, the risk goes down. So, the government has invested in Australia, where we are very lucky in that we get free mammograms when you turn 50, so you can get early diagnosis of breast cancer, if you do have it. So I would urge everyone who is eligible to do it. But for the future, as the population is an aging population around most of the developed countries, the number of breast cancers that are going to be diagnosed each year will increase. And whilst we are quite good and we have quite good therapies for most breast cancers, there are some aggressive breast cancers that we don't have good treatments for. So as I mentioned, the breast cancer that my lab studies is called triple-negative breast cancer. So breast cancers can be broadly classified into 3 main groups, and within each group, there are other subgroups. But for the purpose of today, I will just stick to the simple classification of 3 groups. So the first group, which is the predominant group of breast cancers, and accounts for about 70% of breast cancers, are what we call the hormone receptor positive breast cancers. So these patients have estrogen receptors and progesterone receptors. And the second group of breast cancers, which constitute about 15% of breast cancers, is what we call the HER2 positive breast cancer. So HER2 is a growth factor receptor. Now, the key thing about these 2 groups of cancers is that the hormone receptor positive breast cancers are totally dependent on estrogen signaling and progesterone signaling for their growth and for their survival. And likewise, for the HER2 positive breast cancers, they are dependent on signaling from this HER2 receptor in order to grow and to survive. So for these 2 cancers, because they are dependent on either the hormone or the growth factor, we can have targeted therapy that to starve the cancer just by depleting them, either of estrogen in the hormone receptor positive group or of signaling from the HER2 growth factor downstream. And the treatments are much more specific to the particular cancer type and have much fewer side effects. And because the cancers are dependent, they will die, and we get good therapeutic recovery response rates from them. So for these 2 cancers, we can achieve over 90% of patients have a good 5-year survival. Now, triple-negative breast cancer is called triple-negative because it lacks the estrogen receptor or progesterone receptor or the HER2 growth factor receptors. And it accounts for only about 15% of breast cancers, but it is the most aggressive. And part of the reason is that we don't really know what is driving these cancers to grow and survive. And because of that, we don't actually know how to starve them like we do with the other 2 groups of breast cancers. And on top of that, triple-negative breast cancer tends to occur in younger women. So the median age, they're often women in their 40s who still have younger children and so forth, and of working age, whereas the other 2 groups of cancers do tend to apply to women who are middle-aged. And although it accounts only for 15% of all breast cancers, it accounts for 30% of breast cancer deaths. And part of the reason is because there's a lack of reliable targeted therapy. So these patients are treated with chemotherapy. Now, chemotherapy, I don't know, most of you might have heard of it. It's a bit of a sledgehammer approach. Chemotherapy just kills cells, kills all actively dividing cells, including cancer cells. But there are other actively dividing cells in our body that's required for function, red blood cells, intestinal cells, etc. And so you do get a large range of side effects, and the side effects are not pleasant for chemotherapy. However, about a third of patients actually respond really well to chemotherapy, and they can be cured. But 2/3 of patients will become what we call chemo-resistant. They become resistant to chemotherapy, and whilst chemotherapy might kill the cancers initially, but after a while you get resistant cells that are no longer killed by the drugs, and they continue to grow. And when that happens, when the patients become chemo-resistant, their survival rate's pretty low, and their prognosis is pretty bad. They usually kind of die within about 2 years after they've been pronounced chemo-resistant. And we don't really have good solutions for patients who become chemo-resistant. So one of our interests in the lab is to try and understand how cancers become chemo-resistant, and to try and find ways where we can target the chemo-resistant cells. Now, as I mentioned, about a third of patients actually get cured with chemotherapy. So, and given that there are a lack of other therapeutic options, we thought if we can overcome chemoresistance, then then more of the patients can actually be cured just by giving them chemotherapy, and they don't go through this horrible treatment for no reason, for no outcome, no positive outcome at the end. So, as you probably know, almost all cancers arise from one cell that has gone rogue, right? So instead of dividing when it's meant to divide, and then stopping, and then forming, differentiating to form the correct architecture of whatever organ they're in. So in the breast, they're the ducts that carry milk, and it's the ductal cells in the breast that become cancerous. So the one cell becomes rogue, and it doesn't stop dividing. So it just keeps dividing, and it forms a lump instead of a duct. So we get what you usually find during diagnosis, a lump in the breast. But so cancer occurs by the time it starts, and by the time usually when you feel the lump, it's probably been years. And whilst it's come from one cell, and initially all the cells are identical, they're just divided up, they're replica of each other. Over the course of time, as it grows, it acquires new mutations. So rather than all the lump being just these blue cells that was the original cell, you've now got cells that have acquired new mutations that are different from each other. So you get this yellow cell, this pink cell, this green cell, they've all got a slight different genetic makeup. And when you treat a cancer with chemotherapy, almost all the cells will die, and you get shrinkage of the tumor, which is what your clinician measures, tumor shrinkage. But if you do have one cell, for instance, this pink cell, that's got a mutation that allows it to escape the killing effects of the drugs, then once chemotherapy is completed and the drug's no longer there, it is still alive, it is still a cancer cell, it will start to divide and grow and multiply. And this could take 2 years or 3 years, and often you'd see what oncologists will call a recurrence. Cancer is initially treated, cancer is gone, but in 2 or 3 years' time, you get a recurrence, a new cancer. If you're lucky, the cancer stays in the original place where it's arisen. But if you're not lucky, then over the course of these 2 years or 3 years, they will metastasize, either through the blood vessels or through the lymphatic vessels, as Natasha pointed out, and then treatment becomes so much more difficult. So the questions that we initially set out to answer are, how do we recognize patients that will become or are already chemo-resistant, some of whom are at the time of treatment? And in other words, we want to find biomarkers that will tell us which patients are likely to become chemo-resistant. And the second question, of course, is how do we treat these particular patients that have these biomarkers? which leads us to therapeutic target identification studies. And most of these studies come by trying to understand the biology of the cancer and trying to understand what genes, what proteins make them survive better, make them grow better. So we've been doing breast cancer research for over 20 years. And over that time, we learned more and more about the biology of the cancer and so on. And about 5 years ago, we've been lucky enough to strike upon a protein. So this protein is what we call a kinase. There are lots of different kinases, but it's a particular kinase. And kinases modify the functions of other proteins. So we discovered that this kinase itself, say on this pink cell here, is itself aberrantly modified in cancers. So we've been able to develop an antibody that specifically detects the aberrantly modified protein in cancer cells. And using this antibody, we were able to show that about 25 to 30% of triple-negative breast cancer patients have this marker on it. Now, we were also lucky enough to find that this particular cancer, this particular protein with this aberrant modification drives chemoresistance. And I keep pressing the wrong thing. So the way we went about to test our hypothesis that having an aberrantly modified protein drives chemoresistance. So when I have in my slides active protein here, it means it's active in driving chemoresistance, right? So we took triple-negative breast cancer cells and we engineered in one portion of the cells an active form of this protein. And because we know the mechanism by which it is active, we can also engineer into another group of cells an inactive form of the protein. And we treated both these groups of triple-negative breast cancer cells with chemotherapy drugs and to look at what happens when it has the active protein and what happens to the cancer cells when it has an inactive form of the protein. And so in these graphs down here, we've treated these cells with a whole range of chemotherapeutic drugs. Taxol and cisplatin are two that are commonly used in treating triple-negative breast cancer patients. And so we measured the percentage of cell death when we treat these cells with the chemotherapeutic drugs. And on this axis here, we have increasing dosage of the drug. So it might be pretty obvious that the cells that have the active form of the protein do not die much, even when we go to really high concentrations, high doses of the drug. Whereas the cells that have the inactive form of the protein die, almost 100% of the cell. We can achieve 100% cell death when they carry the inactive form of the protein. So we thought, well, this gives us a potential therapeutic window. If we can somehow, in the cancers, develop a drug that we can give to the patients and turn this active protein off into an inactive form of the protein, then we can continue to give them the chemotherapeutic drugs and they should respond and the cancer cells should die. So we screened a library of chemicals. They're just small little chemicals that pharmaceutical companies have developed and medicinal chemists have developed. And these chemicals are known to have properties that can bind to proteins and interact with proteins. So the way we did this was to take the triple-negative breast cancer cells that have the active form of the protein, and we add Taxol to these cells, the chemotherapeutic drugs. And as I mentioned, most of these cells will not die. They survive the Taxol. And then we add together with the Taxol each of these different chemicals from a library. We screen actually 20 million different chemical structures, right? And to look for ones that can enhance and restore cell death to these cancer cells. So what we found is, so I've shown you, I think we have eventually found about 8. So it was after screening 20 million compounds, we found 8. And I'm showing you the data from 4 of the best ones. So when we look just at the target activity of the protein itself that these drugs bind to, right? So if we do not add any of the compounds, the protein is 100% active, and each of these different compounds reduce the target activity to some extent, by about half to about 2/3. But when we measured cell death, we were really encouraged by this, because so if we just add Taxol alone to the cells and measured cell death, it's very little cell death. But we add Taxol plus one of these compounds, we can essentially sensitize these cells to die in the presence of Taxol. So we are very much encouraged by all of this. And so just to kind of summarize, so over a period of about 5 years, we've taken this program from identifying a gene or protein that drives chemoresistance in about, a third of triple-negative breast cancer patients. It may be more than that. We've come up with a way, come up with a way that we can switch the activity of a protein to driving chemoresistance, to being inactive, and therefore the cells responding to chemotherapy. And we've developed a diagnostic test using our antibody that recognizes the specific form of the aberrantly modified protein, so that we can identify patients that would benefit from any drugs that comes out of our program. But, so the little chemical compounds that we have, these guys here, right, are not really drugs that we can put into patients. So there's going to be, going forward, there's going to be a lot more work to turn them into drugs that we can put into patients, right? So as you can see here, they're only effective in reducing the activity of this protein by about half. And we don't achieve 100% cell death, and we have to use quite high doses of these chemicals. And really, you don't want to use high doses of any chemicals when you're treating your patients, because it's inevitably going to hit some other proteins that you don't want it to hit and end up with side effects. So we're now embarking on a drug development pipeline, which will probably take us another 5 years. But, so proteins are a string of amino acids that fold up, and there are clefts and bits that jump out and so on. And the active site of a protein that carries out its function is usually got a bit of an indentation, what we call a cleft. So I've shown it in this cartoon here. So the chemicals that we currently have are kind of crude. They probably just sit loosely on this cleft. They probably only have 1 or 2 points of contact with the protein, so they can fall off readily, right? They come together and then they don't bind very tightly, they fall off, and therefore they're not as efficient as we want them to be. So we're now working with some medicinal chemists to try and modify and optimize the structures of these drugs. So maybe making them smaller so they can actually fit into the cleft, modifying them so they have more contact points between the drug and the protein, so they bind tighter to the protein, not less likely to fall off, and therefore we can use them at lower dosages for this. And once we're satisfied if we've got a drug that is binding to the protein correctly and efficiently. There's also the question of the drug getting in from your bloodstream into the cells. So we've also got to design the drugs so that they're permeable to get into the cells. And then once we've achieved that, we start the process of in vivo testing, initially in mice, animal stuff. And the first things that we will look for are safety. So you want to give them escalating doses and hope your mouse does not die. If the mice die, then we have to go back to the drawing board and modify the structures again so that they're not so toxic and don't kill the mice. And the second thing that we will look for is efficacy. So we have developed mouse models of triple-negative breast cancers that have the specific aberrant protein in them. And we'll test the drugs for their ability to reverse chemoresistance in these mouse models. And once we've got through all of that, then we are ready for the process of starting human clinical trials. And again, we start with safety trials, phase 1, phase 2, usually on safety trials, make sure that we can put in various doses and it's well tolerated, don't have too many side effects. Then we kind of look for efficacy in the cancers. Are the patients now becoming less chemoresistant? Hopefully they will be not chemoresistant at all. So yeah, it's probably going to take us another 5 to 10 years to get there. Anyhow, so I am just going to finish here. I don't know how much time I've taken, but anyway, this is the Center for Cancer Biology. It's on the other side of North Terrace, as Natasha's mentioned. So these are my members of my group who've actually done the work. These are my suite of collaborators. These are my funding bodies that support the work, without which we can't do what we're doing. So thank you very much for your attention. Thank you for coming.
Thank you very much indeed. Questions? We've got quite a vast generous amount of time for questions. I'm sure if you want to ask a question about how things working for a blastoma in children's, I'm sure she's very happy to take that on board as well. Yes, question already.
Thank you, that was excellent. You know, professional female swimmers who have very little fatty breast tissue, is the incidence of breast cancer more or less? Has any research been done on that?
I don't know the exact statistics. I don't know that it's actually been done, but the difference in breast sizes generally tend to be a difference in the amount of fat tissue in the breast. So the breast is made up of cells that form the milk ducts, so they produce milk when you're lactating and carry the milk to the nipples. And then the rest of the breast is largely filled with fat cells, adipose tissue. So the differences tend to be in the amount of fat that's in the breast, and they're not cancer-forming cells. But there is something that is being studied and is coming through the process, is density. So breast density, which is comprised largely of numbers of cells, but also the amount of collagen and fibrous material that's deposited in the breast. And breast density is a predictor of higher risk for breast cancer. And also, I think they are also an issue because mammograms, it's harder to see a small tumor in a very dense, fibrous breast tissue.
Now that you have the shape of the protein of the cancer, is it possible to genetically modify a patient's killer T cells to produce that as an anti-protein to block the cancer instead of having to develop a drug that could do that?
Right. So, anything is possible, and the technology is certainly there and can be developed to the extent that it is. But genetic modifications tend to be permanent and long-term. So proteins do not often have a single function. They do tend to have other functions. And so whilst we know that when it is modified, so the modification is the presence of an extra phosphate group, for those of you who are chemists and are curious. So the presence of that phosphate group gives it an additional function, which in a cancer cell happens to protect them from chemotherapeutic drugs. But we don't really know at this stage what other normal functions this modification does. It's not present a lot in most normal tissues. We don't see it very often. But I don't think it would, it would have survived this particular modification, this particular structure of the protein would have survived evolution if it didn't also have a normal physiological function. So I think to make a genetic modification, yes, in the same way we engineered the cells that have the inactive form of the protein, but that's going to be permanent, and we don't really know at this stage whether if we do that permanently, whether that's a good thing or a bad thing for the person.
Thank you very much. It must have been very exciting to find that protein that would actually turn off the effect of the chemotherapy. You're working on that. I presume there are other proteins that are doing the same thing, but you're focusing on one. Are there other teams working around the world looking at other proteins and the similar pathway that they'll have to follow?
Yes. I mean, it's, the study of therapy resistance has grown immensely, and it is not just focused on chemotherapy. So, for instance, the HER2 positive cancers have very good targeted therapy in the form of Herceptin and other newer versions of Herceptin, but those patients eventually, the HER2 growth factor receptor that the drug targets continues to mutate, and eventually there'll be a form that is no longer able to bind the drug that targets it. And pharmaceutical companies keep coming up. So, they sequence the patients that become resistant to Herceptin, and they look at what mutations has occurred. They then modify the structure of Herceptin to a new drug that will fit better the shape of the protein when it's mutated to a form that's resistant to Herceptin. So there are lots of groups all over the world that are studying chemotherapy resistance, other forms of therapy resistance. We have not, I don't know of any other group who's studying the same protein that we are, so we've been fortunate in that sense. We are also, and so as part of the curiosity thing and understanding the mechanism of how this protein works, how it's driving chemoresistance, we're looking at other proteins that it interacts with and that functions downstream of it to carry the message to the cells to grow, to become resistant. And we have identified a number of other proteins that is part of the cascade of the pathway, and we're trying to study them as well and looking at opportunities for targeting them, because as we've learned from other forms of targeted therapy, that the cancer cells will find ways of mutating that therapeutic target to become resistant. But the new thinking is that if you can target multiple proteins along a pathway, you've got a much better chance of avoiding resistance, because the cancer now has to have mutations to protein A, B, C, D along the pathway. So that's part of our ongoing work as well, is to understand what's downstream of this protein.
Breast is a body part that serves no purpose past few months after childbirth. So why don't patients and clinicians advocate mastectomy, especially in the group of triple negative, 15% of breast cancer?
That's a good question. I think, I don't know. I think most of us, it's like amputating a part of your body, right? And I think a lot of people have issues with that. Some, those who have BRCA1 they know they're going to develop breast cancer, and they've taken the action to have a double mastectomy, right? I think it's a good question, but I don't know that there's a good answer to it. Yeah.
It certainly gives one a sense of how long it takes from the discovery of the protein. Then you've got to think about the shape of it. Will it fit into that little cell?
Yeah. So we worked with some crystallographers who can make crystal structures of the protein together with the drug, and so they get an image of how it fits and where it's making contacts and stuff. And I'm not a chemist or a crystallographer, so I tend to have to work with a lot of people, the medicinal chemists to modify the structures, the crystallographers to make the crystals with the structure, to look at what they look like when they bind. Yeah, it's a long process.
Thank you very much indeed.
Thank you.
I'm going to thank both speakers in one moment. I just need to find a little gift for them. I think it's here somewhere.
Oh my gosh.
We have a little gift, I think, somewhere. Lovely, thank you. Thank you very much indeed. I wonder if you'd both come forward. We've got a very obviously labelled gift for you both. It's easy to identify, not so easy in your work, but it's easily identified. But we do want to thank you very much for what you're doing. And mostly informative, really important and very revealing session. And thank you very much for your time. That's, as I said at the beginning, the last of our successful ageing seminars for this year. We look forward to seeing you in 2023. Travel safely home, and we'll see you in 2023. Thanks for your attendance.